Hi, everybody. Welcome to Crop Protection Network. I'm Carrie Harmon, your friendly neighborhood plant pathologist, and I'm based at the University of Florida. I run a diagnostic lab that serves hundreds of plant folks every year. Before we get into the specifics of diagnosing diseases, I want to emphasize why we bother. We have got to have accurate diagnosis for effective management. So we're going to use this day to step into practical plant pathology. We're going to summarize what we call triage and diagnosis and turn it into desktop diagnostics, or DIY, just for you. This is really about looking at a sample or looking at your field and thinking about what you know, what the sample or your field and the symptoms say to you. Then we'll dip our toes in the specifics of management and a few apps and other tricks and tools that might be useful for you. There's a TV angler who used to talk about choosing lures by saying, and this is a bad quote, but the first thing a lure has to do is catch the fisherman. I'm obviously not much of a fisherman, but in plant problem management, the first thing we have to do is notice there's a plant problem. Once we've got that, the rest is really just process. And it's a process that you can do from the dashboard in your car, the dashboard of the truck, whatever you are, and from walking in the field. First, we're going to describe the problem so we can figure out if it's likely caused by something we humans are doing, which we scientifically call abiotic, but it's us, or something like a bug or a fungus. Then we can get a little bit more specific so we can pick the right management tools, insecticide versus fungicide. My job today is to show you the process and the tools so you can do it yourself when you need to, and also to tell you who you can call when you could use a little backup. First off, we got to look at the big picture, folks. We got to look at the big picture every day, right? When did the problem start? What does it look like? And how bad is it? These are the first things we really have to identify and get kind of specific about it. So the first test of whether you're dealing with a disease or a pest is to determine if it's nearly all the plants or just a few. If it's nearly all the plants, then the likelihood is really low that it's actually a disease or a pest. That's because things like bugs and fungi take time to grow to a population that can spread and cause a symptom. Also because they're generally really picky, choosing just one kind of plant at a time. So the next thing we want to think about is timing. If it appeared over just a few days or just since last week when you drove by, then the likelihood again is low that it's a disease or a pest for the same reason. It takes a while for that population to cause something that we can actually see. But things like flooding, windburn, nutrition issues, those might take time to show up on a scale we can notice too. So that complicates the timing clue, but the percentage affected will still be high, still guiding us towards abiotic or something we humans have done. So assuming that we figured out by distribution and timing that the issue is biotic, a pest or, a disease of some sort, we now have to determine if it's likely a disease problem or a bug infestation. Remember when I noted that pathogens are picky, attacking one type of plant at a time usually? Well, bugs, like insects, mites, nematodes, may attack a few types of plants at the same time, but the symptoms will be similar across all the plants affected. With bugs, we look for insect feeding damage, leaf or flower distortion, spider mite webbing, and frass and sooty mold, those last two, both of which tell us insects are feeding. Here's where the zoom function on your phone or a magnifier of some sort can be really helpful. We're going to talk a little bit more about that later when we get to tips and tools. Since we're talking about DIY disease management, let's assume we've decided it wasn't nutritional or some other abiotic thing that we did as our human input into the crop, or that it's a bug problem. But we're going to talk about diseases today because, well, I'm a pathologist and I like to talk about disease. To get to a management tool, we've got to decide if the disease is likely viral, bacterial, or fungal. Generally, honestly, I hope it's fungal because we have effective tools for many fungi if we catch the disease early enough. Though for all diseases, prevention, and I'm going to say this more than once, prevention is the cheapest and most effective management tool. Fungi and bacteria tend to start and spread in wet areas, places with shade, near the sprinklers that keep nearby plants wetter than the rest of the crop or create splash zones, low spots where water might pool, or where plants are packed together, maintaining a humid area within the canopy. So we tend to see these really take off when we get canopy closure. Viruses, on the other hand, often get going either sporadically throughout a seedling or transplant type crop, which tends to indicate a seedborne issue, or near wind areas or where insects might gain entry to a crop, say the windward side, or along traffic areas where people might touch many plants along a row. Fungi are bullies. Here's a clue for how to look for what those symptoms are going to be, because they can punch through major leaf veins. They're less restricted in their growth, and so they're going to create round, target-like spots. They can also create tough structures that survive in soil and can be transported across from one field to another from the soil on truck tires or tractors. Bacteria are a little wimpier and wetter, and that's going to be a major clue for diagnosing them. So we tend to see that they cause water-soaked spots with angular edges because they can't punch through those major leaf veins, or starting at hydrothodes and anywhere water collects on the plant, edges, tips, petioles. Virus symptoms, well, they're kind of just weird. Mosaic, spotty, twisted, distorted, and often the newest growth is affected the worst as the virus builds up in the plant, takes over the machinery, and causes just weirdness. So to summarize, I want you looking for patterns, timing, and distribution of the plant problem, whatever it is, and then describe it on the individual plant to key in on abiotic versus biotic and bug versus disease. Remember, if you remember nothing else today, that fungi are fuzzy because they produce mycelia or spores that you can see with the naked eye or with a hand lens. Bacteria are wet because bacteria digest ahead of the cells that they're actually in, so things just look water soaked, really like you spilled a little bit of your coffee on your shirt. And viruses, they're just weird. So here's my favorite way, our first tool of the day, a way to capture symptoms and signs. This little $7 magnifier I get at some big online store fits any phone and has a light so you can use it in the shade house, truck, within the plant canopy. This is really important because hand lenses, while we're all really used to using them, and frankly, they're pretty indestructible, you have to use them where there's light. So you got to turn your hat around because you can't have it in the shade of the brim, and you can't use it very well within the canopy of the plants. This little magnifier, though, lets your phone do a lot of the work, and it can help you decide if that white fuzziness you're seeing is mealybugs or if it's fungal mycelium. So that gets you halfway to solution, insecticide versus fungicide. Pick the wrong one at this point because you can't see it close enough, and you waste money while the problem grows unchecked. But pick the right one, fungus versus insect, and you've got a good chance of managing the problem right now. Totally DIY. So now let's tune our eyes to what to look for to guide us towards biotic problems, evidence of the fungi and bugs that might be causing the problem. So we call these signs, not like here's your sign, but sort of. So signs of the pest are going to be the pest itself, whether that's a bug, whether it's sooty mold, mycelium fuzziness, something like smut, which would be a sign of the fungus itself. We'll also look for symptoms. Symptoms are the plant side of the equation, the way of telling us there's something wrong, but they've got a really limited vocabulary. So using the same symptoms for many causes makes it kind of hard. Symptoms alert us to a problem, but they rarely complete the diagnosis, even here in the lab. It's kind of like a cough might be caused by allergies, a bacterial sinus infection, coronavirus, or maybe you put too much hot sauce on your lunch. Symptoms like yellowing and wilting in a plant could be caused by drought or flooding, so too little or too much water, a fungal root rot, a bacterial stem rot, or just larvae chewing up roots or stem. So let's assume we've gotten pretty close. We're pretty sure it's a fungal disease. We kind of know what we've got. Well, now you got to know what to do about it, right? And that's why I do plant problem diagnosis, is I want to know what to recommend for management. So the University of Florida, which is where I am, hires faculty like me to help you find the right solution for your plant problem. Some of us also run field trials of products to ensure the products that are labeled for use at your site type are also efficacious. So not only can you spray it, but should you spray it, because it'll actually work. Labeled for use doesn't guarantee that it works very well, but your extension specialist has access to the science and data where the products have been field tested, hopefully under conditions similar to the ones you're growing in, to be sure that if there's a product that will work, we know how well it will work, how best to apply it, within the legal restrictions and practical constraints like budget, application tech, and supply. So let's talk a little bit about why it's important to know exactly where you're headed when you're picking out a plant problem. In our case, we're going to talk about viruses, then bacteria, and fungi and water molds. So let's talk about viruses. If we're trying to find a solution for a viral problem in the field, we know that viruses are systemic. So once they're in the plant, there's nothing going to cure the plant. You can't prune it out. You can't mow it off. Viruses are there. even if you can't see symptoms yet. So management really has to be about prevention. It's preventing bugs that might carry the virus or preventing virus-infected seed from getting planted in the first place. So keeping the bugs out is important because they can move the viruses from one plant to another by sucking sap and virus out of an infected plant and then moving to a healthy plant and injecting the virus into it. So the start clean, stay clean mantra is really important here. Don't use plants that are questionable and make sure your seed is from a reliable source so that you can heat treat it. Not all viruses can be eradicated from seed this way, but it's one way to decrease the pathogens in your material. Quickly getting on top of insect vectors that do sneak in, that's really important too, because you want to get them before they reach an unmanageable population size. So scouting frequently and sticky cards are going to be necessary to find small populations while they're still manageable. Bacteria, well, frankly, they're also managed best through prevention. If you do find you've got some, you're going to seek and destroy if you can. Get infected plants out and then protect the plants nearby. If you use flooding, or if you have irrigation from a recycled source or like a surface source like pond water, this is especially important because bacteria can be spread from one infected plant to literally everything else. So you want to minimize overhead watering in any areas you're trying to manage a bacterial disease in, as bacteria are easily splashed from one plant to another. There are some biological products that have shown efficacy by either inducing a sort of immune response in the plant or by occupying the same space a pathogenic bacteria might, outcompeting it. But complete coverage is necessary, and it's preventative only. Protection, prevention, that's how we're going to manage bacterial diseases. Once bacteria gain access to the plant, they're protected by the plant, unfortunately. So any contact protectant also requires frequent reapplication and very good coverage to match the growth rate of the plants, because new growth won't be protected. So since we're talking so much about water, splashing and that kind of thing, let's start on fungicides by talking about the water molds. As with the bacteria, you'll always be money ahead if you can remove the infected plants or destroy them or manage that area by just not getting through it, walking through it, tractors through it, before you treat the rest of the plants. You want to get rid of the source of the inoculum if you can, because then your applications work better on the plants where the inoculum load is lower. If you've had a Pythium or Phytophthora problem, which are the water molds, or some of the downy mildews, make sure you're monitoring recycled water, whether from a surface pond or any kind of tanks. These water molds have swimming spores that are attracted to plant growing points. That's how they find them, especially roots. And also, they form survival structures that are nearly impossible to destroy completely. So destroying the infected plants is still the first step in managing an ongoing disease issue. And of course, target the plant part for best management with the least cost. You're going to drench for root rot. You're going to spray foliage for foliar blights, making sure you're only expecting systemic movement from systemic products. And remember, most systemics really only move up and not down. The rest of the fungi, we're going to talk about having somebody to back you up for identifying which are true water molds and which are the true fungi. But most of the fungi fall into what we call basidiomycetes and ascomycetes. Don't worry about what they are. That's for your lab to tell you, and that's no big deal for you in the field. Just knowing that they're true fungi tells you that they are treated differently than the water molds. A discussion of all the effective products for these true fungi, it's a course in and of itself, and your local extension folks are going to give you the most effective products for your area anyway, and stay current. So I'm not going to go into them specifically, but I want you to remember that you generally can't spray your way out of a problem cheaper than preventing the problem in the first place. And a perennial plant or a repeat crop will likely need perennial treatment during disease-conducive conditions, because fungicides are never going to get 100% of the fungus. There's always a chance the problem will reoccur as soon as conditions are right. But if you stay on top of it and plan for the sprays and try to stay clean when you can, you'll be able to protect, prevent, and then treat at a lower cost and with better results. So we're still in DIY. I want to remind you that every state has resources developed by local experts for local crops. The Crop Protection Network is going to connect you to these resources formatted for regional or national use, but every state, you're going to have it formatted for your state. And that's partly because pesticide labels are state-based, but also because conditions change. What crop is grown in one area on certain soil, it's going to look a little different when it's grown under different conditions, windier, different soil, different irrigation practices, different pH. Those things are going to change from state to state, region to region. So your local folks are going to be able to tell you the most important stuff. So I was asked to talk to you a little bit about other tools that you can use. Because I do disease diagnostics, I got to tell you, we don't have a lot of tools to replace people, but there are several apps on the market to provide some additional diagnostic and decision-making help. So I want to tell you a little bit about them and a few things to consider when deciding to use one. First, does your app connect you with your local or regional experts, or at least their expertise? Meaning, is this something that was developed in another country or another region, and is it going to have application to where you are? Two, does the app focus on your type of crop or have a section that does? If it's mostly greenhouse crops and you're doing field stuff, might not have the same application. Also, the images of the disease and the symptoms of the pests that you're dealing with might look really different out in the field than they would if the photo had been taken inside a greenhouse, for instance. And then, since we're talking about photos, can you take really good pictures so the app's artificial intelligence can take a look at what you've entered for your crop and can accurately recognize the symptom or pest? A lot of these apps will let you take a picture of what's going on in your field and then run it through an algorithm of other images, comparing it to those and come up with the best possibilities for you to consider. Those are really good for helping you zero in on what the issue might be. I've put a few examples of some of the DIY type apps that you can apply to your situation out in the field. I have no involvement with any of these. My mention of them is no more than providing examples. Mention does not equal endorsement. There you go. You got all the fine print real quick right there. But apps like these can be helpful for recognizing the type of symptom, especially with foliar issues. Insect issues are also much more likely to have a correct ID with these tools, simply because you can see the bug, right? Whereas with diseases, it's rare to be able to clearly see the pathogen. The other thing is with diseases, the symptom has to be a really distinct one to be able to get an accurate diagnosis. But these apps can totally help steer you towards a group of possibilities to consider for either consulting an expert or doing a little more research yourself. All right, so we did the DIY, the do-it-yourself. Now let's talk about CAP, when to call a professional. We've talked a lot about how to figure out what you're dealing with in terms of insect versus disease and fungi versus bacteria or virus. Then we covered what we need, why we need to drill down to choosing the right management options, keeping in mind that a good offense is still the best defense. It's always going to be more effective and cheaper to exclude the pests and diseases than to try to spray our way out of a disease outbreak. But disease management is part of crop management. So we've got to know our chemical options just so we can be prepared in the event we have to deal with something. We work within the boundaries of virus, bacteria, and fungi, and water molds, but I group them in the fungi too. And within the fungi, we've got to know which kind to be sure we're choosing the correct mode of action and application technology for the situation. If you're seeing something new or just aren't sure if that leaf spot is likely to spread, now it's time to CAP, call a professional. Determining the specifics might be a bit much in the field, which fungus it is, whether it's a water mold or not, whether there's more than one thing going on. Even with the magnifier and the apps backing you up and a good photo, you still might need to call a professional at this point. So, if the previous stuff that we've talked about seems like a lot to sift through, I want you to know about some of the folks that have your back. The National Plant Diagnostic Network is a consortium of all the university diagnostic labs across the country. Folks like me, all over the world, or all over the country. And actually, we do have folks all over the world through the International Plant Diagnostic Network and through other groups as well. There are diagnostic labs pretty much everywhere that you grow plants. But within the National Plant Diagnostic Network, if you go to npdn.org, you will see this map on our first page. Clicking on your state on this page will take you straight to your local experts, your local lab, your friendly diagnostician. Accurate identification of the problem is of prime importance in effective and cost-effective plant problem management. Most of these lab folks work closely with their crop-specific extension specialists, keeping the experts informed about what's popping up in their crop of interest and passing along research-based management information from them to the sample submitter. It's a two-way street that ensures we all keep headed in the right direction. We're all on the same team after all. If you did need to find a lab and interact with them and potentially submit a sample, you can find contact information, prices, submission forms, and sampling instructions, which can be really helpful, all for your local lab by clicking on one of those pictures of your state. This is an example here on the screen from my lab here in Florida. Most labs have a similar form and a similar format. They all look a little bit different, and of course, the mascots are going to change. But the information on the sample form is really important. A short run through, I'm not going to tell you every little bit of it, but a short run through of the really big highlights will help us think about sampling as we proceed through the rest of the program. So to start, you never want to send a lab anything dead. We can't do postmortem. This isn't like CSI. Postmortem analysis doesn't work except that we can tell you that the plant is dead. It's not particularly useful for any of us, right? So it'd be a waste of your time and money to send it to the lab. The best time to sample is when you first notice a problem and before you spray anything. But if you can't get out there before you spray, it's still okay to send a sample to the lab. We can usually still find the cause of the problem if you send us something after you spray, but the fungicides will knock back some of the pathogen growth, most likely, and diagnosis might take longer. However, you're getting an additional piece of information, and that is whether or not what you sprayed is effective. Plant photos, whatever you see, helps your diagnostician or your extension specialist be there in the field with you when they can't actually be there in the field with you. So really good photos of the field and then of the area of concern and then a close-up of the plant and all the plant parts really helps. When a lab receives your sample, usually you'll get an email receipt or a phone call or something. Somebody will let you know that we got it. In my lab, the software we use is once we log everything in, you get an email that says, hey, we got your sample. Here's all the information we have about what we entered. Did we get it right? You can then hit reply and send those photos to us or any other information that you think would be useful. You can also tell us if we spelled your name wrong or got the county wrong or something else within the plant sample information. You can also send us your spray application notes and you can ask us questions. And as for sample turnaround, because timing is of the essence here, you can expect to hear from the lab most of the time within a week, sometimes much shorter than that. And if you need to know fungus, bacterium, virus right away, you can usually call the lab and ask them to do kind of an on-the-spot hypothesis. It'll have to be backed up by some of the lab tests to make sure that we've given you the right diagnosis, but we're always happy to talk to you in the field because then we can get more information about what's going on. Everybody gets to a better diagnosis for better management later. We want to get you a quick and accurate diagnosis, and you can help with that by sending good samples, good information, and getting them to us quickly. This is not the time to use the slowest shipping process or to let something sit on the dashboard of your truck. So that was a lot. I know that was a lot, but I hope you found a few resources you can tap into the next time something is looking unhappy in your field. You can always look me up or your local lab folks up when you need a little backup. So thanks for attending this, and may your plants be perfect and profitable.